So there are three main categories of molecular models that we are going to cover. We're going to cover carbohydrates and proteins and lipids. And there we go. So my chat had disappeared, so now it's back up. Okay. The first thing to think about with a molecular model, with molecular models, are what are you building with? So what are your atoms? And so what we use a molecular model kit, and so okay. So I have a kind of a strange question. Can you see me, both me and the screen, both the PowerPoint and me on camera? Yes, okay. Sorry. It's a new Zoom world for me. Okay. Awesome. Okay, that's good. Okay, so when we are building molecular models, we have a molecular model kit. Okay, so if you see a black ball, it's going to represent one singular atom of the element carbon. And it's probably a little hard to see, but there are 4 holes in there. And it's because carbon likes to make 4 non-covalent bonds. Okay. And you'll learn about bonding in lecture. For lab, what we do is we build models with our kit. And so these just represent bonds. The gray sticks represent bonds. So carbon will have 4 bonds. And then we have hydrogen, which is usually tiny because it's a very small molecule. It actually can only make 1 bond, so it has just 1 hole. So if you see a white atom, it's the atom hydrogen. It's representing the element hydrogen. We have oxygen is represented by the color red. Okay. Nitrogen is represented by the color blue. And then we have phosphorus, which is represented by the color purple. And when you're doing, if you want to do this at home, what I usually do is I have a whiteboard. Okay, so, and I actually have like magnets and things that are multicolored, but you could just use pens if you wanted to do that as well. So if you ever have questions about chemistry, I love chemistry, and I just diagram things out on a whiteboard. Not everyone has a molecular model kit at home, but a whiteboard works really super well too. Okay, so in real lab, the first thing we'd be doing is just literally holding these atoms up, and figuring out which element is represented by which color. Okay. Then the second activity we would be doing is to build basic molecules. So when you build a molecule, all of the available electrons, which are represented by bonds, are utilized. So this is called, we're going to build elemental oxygen. And elemental oxygen, so oxygen likes to form 2 bonds, but it's going to end up bonding to itself, and it forms a donut looking. And For the lab exam and for the quiz, you need to be able to identify something by its name. So this is probably the strangest one. Elemental oxygen. Water is more normal. Carbon dioxide, methane. By its formula, you have 2 oxygen atoms. So the way we write that is O stands for oxygen. You'll find that on the periodic table for its elemental symbol. And then a subscript of 2 means that there are 2 of them. And then it has a double bond. So a double bond means that you have 2 bonds between the same atoms. When we create water, we take 1 oxygen, it goes in the middle, and then put 2 hydrogens off to either side. So sometimes, it looks like Mickey Mouse ears or a little bow. You can do all sorts of fun things on Zoom, right? But you can see now these are just single bonds because there's only, well, hydrogen can only make single bonds, but there's only 1 bond between each set of atoms. But so if you were to see a picture of this model, and a hint is that you will see a picture of either these molecules or a functional group or a molecule from later on, you'll have to identify it. You'll have to say, oh, well, that's water or carbon dioxide or methane or glucose. So you do need to be able to identify these models. We'll go over, the second PowerPoint has pictures of all of these as well. And if you ever start getting stuck as to what you're identifying, a good way to start is counting how many atoms you have, because if you were to look at this, some people just go, oh my gosh, I know that, and but I don't know that. Just write down that you have 2 hydrogen and 1 oxygen, and you'll go, oh my gosh, it's H2O, and I know that's water. So like for the next one is carbon dioxide. I thought I had built, but I guess. So carbon dioxide, okay, you have a carbon and 2 oxygens. But again, carbon likes to make 4 bonds. So the only way to make carbon happy and oxygen happy, because our oxygen likes to make 2 bonds, double bonds, is to double bond both of the oxygens, okay? So again, when you're looking, if you get a picture of this model, you might go, I have no idea, because I've seen a few models that have double bonded oxygens, like there's a carboxyl group, and then there's carbon dioxide. But if you just start and you go, I know that black represents carbon, so I have 1 carbon, and I know that red represents oxygen, so I have 2 oxygens. So I have CO2. If you just start there and you write down, I have CO2, then it's a lot more straightforward to say, oh, well, if I have CO2, then I have carbon dioxide. Do the bonds represent the lines in the colors? Yes. So on the PowerPoint, these represent like a double bond. So carbon is going to be double bonded to oxygen and carbon to oxygen. When we get to methane, I I didn't diagram that one out as well. But yes, these are kind of a roadmap as to how you would, this is a single bond and this is a single bond. When we get to methane, which is next, I was not as elegant. Is it just, it looks like a jack. It's, because it's, you know, carbon, and then it has 4 single bonds with hydrogen. So it ends up looking like a jack. And it didn't, so it did not diagram out as well here. So these 4 basic molecules, another question that sometimes we would present to you is we would have pictures of those 4 molecular molecules, 4 basic molecules that I just built. And we might, you know, have pictures of them. And then we would say, and they're labeled A, B, C, and D, and we would say, which one of these is carbon dioxide? And you'll have to pick A, B, C, or D. So you do need to, there's a heavy emphasis on recognizing those molecular models. So the second activity is on functional groups. So a functional group is only part of a molecule, and usually it's part of a much larger molecule. So this R here is the rest, the remainder of the molecule. So for example, we'll have glycerol, and it's going to have 3 alcohol groups onto it. For like this carboxyl, we'll see that in amino acids, we'll see it in fatty acids, and carboxyl groups are very highly reactive. So you'll see these over and over and over again. And then there's usually a lot more coming off over to the end. Okay. So this R just means that there is something else following off to the side. And so you'll also, so when we build, okay, an alcohol, sometimes these are also called hydroxyl. It really, I don't know, you know, chemists, they just, sometimes they should use the same names, and sometimes they do, and sometimes they don't. So this molecule here is hydroxyl or alcohol. You'll see it called both. And it's the same thing. It is an O and then an H. So oxygen, hydrogen, and then this open bond here, because there's the rest of the molecule is out here. So this could be a carbon, it's usually a carbon. It could be, what else it could be? Why do some explanation have groups have pluses and some have minuses? Because some things are charged and some things are not. Oh, so this you're seeing, okay, so you think, so this is actually not a minus. This is a bond, or like an open bond. But some things are going to have pluses and some things are going to have minuses. Amino groups tend to be positively charged, and carboxyl groups are going to be negatively charged, so that they will interact with each other. But these are going to be more just representing that there's a bond here that it's going to be attaching to. So this is building, this is just a building off of it. So you've got a bond and then an oxygen and a hydrogen coming off of it. Okay, so your first functional group is alcohol. Then your second functional group is the amino group. And the amino group is a nitrogen, and then a hydrogen and a hydrogen, And then you have, I'm going to put a longer bond on here so you can see that you have this extra long bond coming off. Okay. So amino groups are very important in amino acids because every single amino acid has an amino group. It will also have a carboxyl group. So if you are taking notes, you need to write down that every amino acid has an amino group and a carboxyl group. And we're going to diagram this out later on. But when we make these, we set aside our amino group and we'll set aside our carboxyl group because these are two of the three components of every single amino acid. Okay, so amino groups are very important, that nitrogen atom. And then carboxyl groups are, I'm just gonna, very important too, okay. So for carboxyl groups, we start with carbon. Okay. And then you need to make this carbon, so carbon, you've probably heard that we are carbon-based life forms, right? Okay. Carbon is very structurally stable. That's why we're built out of carbon. Because carbon likes to form these very stable bonds. They're equally spaced, they're very strong, they're very stable. That's why we can build large beings like us. Okay. The downside of that is when you need to change a molecule, you need to get carbon out of its comfort zone. You need to change, activate, invigorate carbon. And the way you do that is you put it in close proximity to we call them electronegative elements. And these are big burly elements whose electrons have a lot of pull, okay? So some elements are big and burly and their electrons want to pull electrons from other atoms and they just kind of shake things up. And so oxygen happens to be one of those. So if we want to get carbon kind of out of its comfort zone, we put it in proximity to oxygen. If we really want to get carbon out of its comfort zone, we're going to double bond it to oxygen. Okay, so when you double bond a carbon to an oxygen, you're going to kind of activate that oxygen and it's going to be ready to participate in a reaction with another molecule. So then if we are going to add another oxygen and then another hydrogen, we are going to have a carboxyl group that is going to be ready to participate in another reaction and combine whatever is on this side. Sorry, this is another molecule out here, and then we'll have another molecule. It's going to get this carbon ready to participate with whatever is over here. So carboxyl groups are exceedingly important in getting carbon out of its comfort zone so that it will participate in joining together in reaction. Okay. Our last one is methyl. Methyl is just like methane, but you pop off a hydrogen and you get that open bond. Methyl groups are used as tags. Often they turn things on or off. Okay, so sometimes we methylate DNA, and it just kind of sends up a flag and says either I'm active or I'm inactive. So we have methyl groups kind of here, there, and everywhere. Okay, so those are your 4 functional groups. If we were in a real life lab, the next step would be building a glucose molecule. We're going to jump over just a sec to a whiteboard because we need to talk for just a second about carbohydrates. So carbohydrates are roughly 25% carbon and 50% hydrogen and 25% oxygen. Okay, so they have that ratio. So if you look at the models, they're going to be roughly 25%, what color? That is carbon. Black, very good. 50% white and 25% red. Very good. So when you see a model, the color ratio also gives you a hint at what you're looking at. Okay. We have single sugars called monosaccharides, okay? And they are what are called cyclic structures, meaning that they are going to have a ring in them. And they have the formula C6H12O6. For those of you that may have had chemistry or something else, if you see the formula C6H12O6, does anyone know what that formula represents? What molecule? Commonly known as? Nope. Okay. So it is known as the molecule glucose. But it's also the same formula for fructose and the same formula for galactose. These are called isomers. They have the same, if we were to build glucose, fructose, or galactose, we would pull out 6 carbons, 12 hydrogens, and 6 oxygens. But we would put them in a different arrangement to build them. But they have, if you then took those models and you broke them apart, you would have no idea which one of those you had built because they're built with exactly the same elements. Okay. So that's kind of interesting. So I will show you how we make glucose. And glucose and galactose are exceedingly similar. We would never ask you to identify the difference between those two because I'll show you where they're different, but it's a very subtle difference. Glucose and fructose are actually quite different in structure. If we were in real lab, we would expect you to be able to identify those. I did not see a picture of fructose in your lab exam review, so I don't think that's in there, but I will show you the difference as well. When we eat, like if you have breakfast this morning, okay, let's say that you had a bowl of cereal, okay. If you had a bowl of cereal, you did not eat glucose, fructose, or galactose. You ingested something called a disaccharide, which is 2 of these put together. And there are 3 of them. So what you might have had is maybe you had milk, okay, on your cereal. And so milk is made up of lactose, okay? We'll see, okay. All right, okay, so back to disaccharides. Okay, so if you had milk, and some of you could be lactose intolerant, and that lactose intolerant individuals lack the enzyme to break down lactose into glucose and galactose. Okay, and so if you buy Lactaid milk, it's had that enzyme added, and so lactose has already been broken down into glucose and galactose, which is why it tastes sweeter, because now instead of a disaccharide, you have 2 monosaccharides. So your taste buds are tasting 2 sugars instead of just 1. If you had, you also would have ingested sucrose, which is glucose and fructose that have been combined. And then we have, oh, the last one, sorry, is if you have a beer this weekend, assuming you're over 21, you would ingest maltose, which is glucose and glucose that have been joined together. Okay, so those are disaccharides. So 2 monosaccharides put together. Now, how do we put 2 monosaccharides together? If we want to put monosaccharides together, that process is called dehydration synthesis. So that is a monosaccharide plus a monosaccharide, and you would get a disaccharide, and you would produce a water molecule. Okay. Well, you had breakfast, so you ingested a disaccharide. Okay, so you had breakfast, so you want to do the reverse in your small intestine. Okay, because if you want to absorb what you ate, you need to do the reverse. And so in your small intestine, you want to do hydrolysis, which is where you break down a disaccharide, and then that is going to go into a monosaccharide, and then another monosaccharide. But in order to do that, you need to consume a water. Okay, oops. Because you would need to, and I'm going to just about show you a video on how this all works. You have to break this water and put it back into this monosaccharide and put it back into this monosaccharide to replace the molecules that were lost when you broke it apart. So you have to put one in here and put one in here to make 2 whole units, okay? Because you had 1 unit here, and if you're breaking it apart, now you're gonna have open bonds. Molecules don't work that way. You've got to put them, fill those bonds back in with something. And that something is the hydrogens and the oxygen that's going to come from a water molecule. So if you're going to break something big apart into little units, you're going to need to use the molecules from water. So you're going to consume water and use those atoms to make whole smaller units. If you're going to use smaller units and squash those together and to make something bigger, then you will produce water because you've got to make space to put those together. Okay, so I'm gonna go back to, oops, my Zoom bar is kind of in the way. This one, and back here. Okay, so I'm going to clean this up a little bit. Okay. Now I'm going to, yeah, because I have to show my sound. We'll go back to this one. Okay. So in this video, it is going to show you how to make a glucose molecule.
In this video, we're going to talk about the bonds that make up macromolecules. Each one of them has a unique type of monomer and a polymer name. So we want to focus on the macromolecules that we've talked about here, but how do we build those polymers, and what are the names of the bonds? Each one of these has a specific name. And so, brought to you by Curious Marineland. So when you actually see this right here, and actually that, and I do have a video for lipids, so I'm going to go in order, and we're going to talk about what exactly does it mean to actually add a molecule specifically for carbohydrates, lipids, proteins, nucleic acids. And here are the bond names that you're going to hear in this video. So what I've done is 4 short videos. I've combined them. I'm going to go in this order, and we're basically going to talk about how these are made. I'm going to use models, and I'm also going to use some pictures. So this is just my intro to that. So stay tuned, and next we're going to talk about carbohydrates, all right, and then lipids, proteins, nucleic acids.
And the only type of bond in particular that we're going to be talking about are the peptide bonds. So you don't need to actually know like the names of the bonds like glycosidic or ester until we get to the peptide bond.
So what do you see? I see 1, 2.
Okay, so he has built 2 glucose molecules. So this is a singular glucose molecule, and this is a singular glucose molecule. And so each glucose has 1, 2, 3, 4, 5, 6 carbons, and 1, 2, there's another one here, 3, 4, 5, 6 oxygen, and then the 12 hydrogens. And then what he's doing is just by convention, they are arranged this direction, and we number them. There's a whole set of rules in chemistry on how we number things. So when he's counting them, it's just by convention, it's how we number them. And on the next slide, I'll show you kind of the same thing in diagram form. You do not need to know that the 4th carbon and the 1st carbon are the ones that combine in dehydration synthesis. This is just showing you the process. You do need to know that the process name is dehydration synthesis, and that you will produce a water, and that you produce a disaccharide. You don't need to know which carbon or which oxygen and hydrogen, okay?
2, 3, 4, 5, 6 carbons. I also see 1, 2, 3, 4, 5, 6 carbons. So I'm going to arrange these carbons so that the 4th one here is going to interact with the first one here. And this, when it's combined, will be a disaccharide, which is a carbohydrate. So if you notice, look at the model. Here's the linear form of glucose. Here's the cyclic one. So you see 1, 2, 3, 4, 5, and 6. So 4 of this one will interact with the 1 of another one. So let's look at it. Here's glucose and fructose that makes sucrose. Here's glucose and another glucose. That's what I'm going to demonstrate. So you'll see 1, 2, 3, 4. So here's 4 right here. And here's 1. And this right here is going to be a bond, AC. And this is the bond that connects one carbohydrate, a monosaccharide, to another. And this is called a glycosidic linkage. Glycosidic linkage. So I have this little purple bond just to show what's going to happen. Typical dehydration synthesis. So dehydration synthesis. So when these molecules combine, some water has to come out. So let's do this. So here's the OH right here. And then I'm going to take off here. You can always show water being produced, and you'll see this in all the videos I do for this dehydration synthesis with the molecules. So here it is.
He actually makes a mistake here. He
This
should have left that
is a
oxygen
disaccharide.
on. That's
In particular, it's
left an
actually
oxygen
maltose,
here.
which is a sugar
So
you see in
he
milk
actually makes
sugars.
a little bit of a mistake in the
So
video.
that's pretty much the first one.
So
I'm going
this
to do a video
oxygen
for
should have stayed in
lipids
here.
and proteins. For nucleic acids, we're going to do like more of the PowerPoints, but I'll also show you what the model actually looks like. So that's the first of a couple. And the whole idea is build a molecule or break it. Biosynthesis or digestion in all essence. All right, next one video is coming.
But overall, the video is pretty good. But he does make that little error, and that he should have left the oxygen. And I'll show you that on the slide. Okay, we'll go back to here. Okay. Oops, I need to share this back. Okay. So again, in dehydration synthesis, we combine a monosaccharide plus a monosaccharide and we produce a water. The same process occurs whether it is for monosaccharides or if it is amino acids, or if you are combining fatty acids onto a glycerol molecule. Whenever you are putting two, what you call these macro or large molecules together, it is the same process. And hydrolysis is the same, breaking them apart. Everybody likes common things. We are going to talk about this peptide bond, how many there are, and how to identify them. So we're going to actually switch back to a whiteboard again. Okay, so there are 20 amino acids that we use to build proteins. And a protein is made up of polypeptides, and a polypeptide is amino acid after amino acid after amino acid, and do that for 100 to 1,000 times. But in lab, we are concerned with what is an amino acid, what is a dipeptide, and what is a tripeptide. Okay, so those are going to be the choices. Ooh. Okay, so we have amino acids, dipeptides, and tripeptides. Dipeptides are made up of 2 amino acids. Tripeptides are made up of 3 amino acids, kind of like a tricycle. Okay. an amino acid is a singular building block. Okay. So it does not have any joining bonds. It does not have any peptide bonds because it's just a singular entity. Now, if you put amino acid plus amino acid, then you have a peptide bond. So if you get amino acid plus amino acid to form a dipeptide, what links them becomes the peptide bond. So a dipeptide, where you combine these two, has how many peptide bonds? So guess? 1. Very good. Because you have one bond that's going to link them. So now with a tripeptide, where you have 3 amino acids, so now we have a tripeptide bond. You have how tripeptide has 3 amino acids. So you have how many peptide bonds? 2. Very good. So an amino acid has 0 peptide bonds because it's just a building block. A dipeptide has 1, and a tripeptide has 2. Very good. Okay, so there we go. So now we're on to the building block of a single amino acid. Okay. So a single amino acid has an amino group. Okay, so that's the amino functional group. And then it also has a carboxyl group. Every amino acid has these 2 functional groups. This is the basic side, this is the acidic side. In the middle, it will have a carbon, it will have a hydrogen, this is called a carbon-hydrogen bridge, and then it has something called an R group. This R group can be any of 20 different things. It can be as simple as a hydrogen. Okay, then we'll get something like glycine. It can be a methyl group, we'll get alanine, or it can be a complex. It can be something that's very complex, and it could be like a ring structure. It can be charged, it can be acidic, it could be basic. It gives, the R group is what gives the amino acid its personality, because we need to build some proteins that are going to be enzymes. We need some that are going to be strong and structural to build things like tendons. Okay, we want some that are soluble in water and some that are not soluble in water, some that are pH resistant, some that are not. We want amino acids to have lots and lots of different personalities, and by having different R groups, that's how we accomplish that. Again, that's kind of a lecture topic, but in lab, what you need to know is that this is the amino group. This is a carboxyl group. And then we join it in the middle. Okay, so this is an amino acid. When we combine them we combine them with that peptide bond to another amino acid. And that process of amino acid plus amino acid to make a dipeptide is called, we are making something larger, it begins with a D. Close. So if you go back to monosaccharide plus monosaccharide makes a disaccharide, we have amino acid plus amino acid makes a dipeptide, the process, the reaction. It's 2 words. Dehydration synthesis. Okay. So we're going to produce or consume our water. Very good. We're going to produce the water. And the way we would do that is we would have an amino acid here, and then we'd have an amino acid on the other side, okay? We would take this OH from this amino acid, and then you have to imagine that you've got another amino acid over here. Okay, so this is the hydrogen off of the amino group, okay? Just gonna kind of bear with me, right? That there's another one on the other side. So you take this hydrogen from amino acid number 2, and you can see, hopefully, how you would form H2O from here to there, okay? So you would form a water. This bond now is going to swing over and connect to the nitrogen on the other amino acid. And that's how those are going to connect. So amino acids are made up of an amino group. They're made up of a carboxyl group. When you combine them to get a dipeptide, you get 1 peptide bond. And that is here, okay? So that's what we were showing, is that if you have an amino acid and another amino acid, you take an OH group from one, a hydrogen from another. This bond swings over now to join with the nitrogen, and you get a peptide bond. So 1 peptide bond in a dipeptide. If we wanted to make another one, we would take these OHs off, swing this bond over to make our second peptide bond in a tripeptide. So here again, this slide has like a lot of stuff that you need. Dipeptide has 1 peptide bond, 2 nitrogen atoms. Tripeptide has 3 nitrogen atoms, or you could replace this with amino acids and 2 peptide bonds. Well, that's not what I wanted to do. Okay. Okay, so we are now moving on to our 3rd category of macromolecules, and these are lipids. There are 3 types of lipids. We have phospholipids that are used in membranes. We have triglycerides, which are used in energy storage, and then we have steroids. So triglycerides start off with, this is called glycerol. This is the backbone, the structural beginning. And it has a linear structure of carbons, and then it has these 3 OH groups. And these are the ones that are going to participate. They're going to donate this hydrogen to attach fatty acids to form a triglyceride. No, it still doesn't like doing that. Okay. Okay, so fatty acids can be either saturated, meaning that there are no double bonds between carbons. Okay, so there will always be, for a fatty acid, there will always be a double bond between a carbon and an oxygen. This was part of that carboxyl group that participated in the reaction. And this is called the head of the fatty acid, and I have a picture in a minute. But there's no carbon to carbon double bond. So it cannot fit any more hydrogens on that. This is called the tail, okay? No more hydrogens can fit. So it is saturated with hydrogens. These are solid at room temperature because they will pack very nicely, linearly. Unsaturated fatty acids will have at least one carbon to carbon double bond. And so they could fit more hydrogens. So they are unsaturated. If there is one carbon to carbon double bond, it's called monounsaturated. If there is more than one, it is called polyunsaturated. And I have pictures of those too also. Okay. A triglyceride is when glycerol has all three fatty acids attached. And this is the way triglycerides are. They don't just end up with two, they end up with three. And this is how energy is stored in your body. And you could have one unsaturated and two saturated, or two unsaturated and one saturated. The representation of fatty acids is mainly going to be according to the diet that you eat, because that's how they're stored in your body. They'll store whatever you eat. And the next slideshow, I have a lot more pictures. The next type of lipid are steroids. All steroids have a 4-ring backbone. It's the only structure that we look at that has this 4-carbon ring structure, where the rings are made out of carbon, and there are 4 of them. And then these are all lipid soluble. They can just go straight through your membranes. They are modified by what we stick off of these rings. And so this cholesterol has a very highly branched tail to make it rigid in membranes. We also have our steroid hormones, estrogen and testosterone. They have very small modifications off of those. For your lab quiz or lab exam, the picture is of the steroid backbone. So what you would be looking for is this 4-carbon ring structure. If you see a 4-carbon ring structure, then you are identifying a steroid. The last one for this slideshow, and then we'll do a quick review, is ATP. ATP is an energy storing molecule. You'll learn a lot about it in lecture. I'm not going to go into its purpose in lab. It is made up of what's called a nitrogenous base. It's the only molecule besides the amino acids that you'll see nitrogen in, but these are ring structures, okay? But the other giveaway is it's the only molecule you'll see phosphorus in, phosphate, okay? These are phosphates. This is the molecule phosphorus. It's purple. If you see purple, it is ATP. Go, thank you, yay. I am so happy to see this on a lab exam. It is the only molecule that you will see purple in. And so this is the molecule ATP. Okay, so I'm not going to spend a lot of time on that one. Okay, so I'm just going to pop over to, okay, so we do have the test review to go over as well, but before we do that, do we have questions. So questions for today's quiz. So you will take that before 10 p.m. today. Yes. Any questions before we go over? So there's 10 questions and you have, well, there's actually 9 questions, but there's 10 answers. One question has 2 parts in 10 minutes. And there's a countdown on the quiz. So that's I think the hardest thing to get right on the first quiz is just the timing. So just make sure that you are keeping an eye on the timer and make sure and answer all the questions. Okay. And I'm pretty sure that it's 5 on the microscope and then 5 on organic molecules, if my memory serves me correctly. Okay, so the quiz comes from anything we talk about. The lab exam comes from these lab exam files, as well as anything from your lab manual. Okay, so there are, where did it go? Okay, there are like questions on pages 15 and 16 that you can review as well. But the majority are going to come from these lab exam files, which is, so I'll have to think of, I'll probably like put them up. I'll probably put these our recordings up maybe between the quiz and the lab exam, but I don't think I can have them up during lab exam week. So I'll have to think about that a little bit. Okay, but we'll go back to this. Okay, so if for something like this, we have black represents the element carbon, blue is nitrogen, purple is phosphate, white, hydrogen, and red, oxygen. Very good. Okay. And then we have identify these molecules. Need to make this up. So A is, we call it elemental. Yep. B is carbon dioxide. C is, I know some of you guys are in a different order. C is water, and D is methane. Okay. These are more from lecture, so we don't have to worry about that. Okay. You guys can do that one, I think, on your own. Okay, so methyl is letter not D. It's very similar. Yep, it's A. Very good. Amino group is so the amino group is going to contain nitrogen. Yep, so amino group is group D. Hydroxyl is group It's also known as alcohol, B. And carboxyl is the large guy at C. Very good. Okay. So yeah, so know your molecules, know your functional groups. Which two of these are components of amino acids? So which two letters? D is correct, that's for one. And then what is the other one that is a part of every amino acid? So the amino group, and there's one other one, C. And what is the name of C? You don't remember? Yep, carboxyl group, very good. Okay. Which one of the functional groups is found in the glycerol molecule? We didn't cover that a whole lot, but so glycerol has 3 carbons, and then it has a bunch of hydrogens, and then it has 3 alcohol groups on the bottom, 3 OHs. So identify this organic molecule. It has an amino group over here. It has a carboxyl group over here. Has a carbon and a hydrogen, and it has another hydrogen here. It is known as not a monosaccharide because it has nitrogen. So the nitrogen, yep, an amino acid. So that nitrogen is going to be your key indicator that it is an amino acid. And there's just one nitrogen. So that would indicate that it is just one amino acid. Now this one has 1, 2, 3 nitrogen. So it is it's not an amino acid, it is a tripeptide. Very good. Okay, so if it is a tripeptide, how many peptide bonds are there in the molecule? 2. Okay. Now, this is more difficult in a diagram than it is in real lab. So it says, is the indicated bond a peptide bond? Okay, so peptide bonds always form between carbon and nitrogen. Okay, so it passes the first test. Okay, but it doesn't pass the second test because the carbon has to be attached, it has to be an activated carbon. So it has to be a carbon that's attached to a double bonded oxygen. This carbon would never have enough energy to participate in a reaction. It would be too happy. It'd be complacent. It'd be like, I'm not doing that. I like it. I like life just the way I am. I'm not gonna change. But this carbon, because of oxygen, has been activated, so it will participate in a reaction. So the bond is always between carbon and nitrogen, but the carbon has to be active. So this is going to be one of your peptide bonds. And then the other peptide bond is this one between carbon and nitrogen, but the activated carbon. So we have one peptide bond here, and then the second peptide bond is here. Okay, so we do So you have one here, and then you have your second peptide bond there. Okay, so two peptide bonds, always between carbon and nitrogen, but that carbon has to be activated. It has to have that, because it used to belong, it's the same one that belonged to the carboxyl group. Right, because one was, they're all, if we had them on a table, they would all be arranged the same direction, with the nitrogen like on the left and the nitrogen on the left and the nitrogen on the left. So it's this carboxyl to this nitrogen and this carboxyl to this nitrogen. So it's always going to be in the same arrangement as long as you have your nitrogens like on the same direction. You know, we could flip this over on this other way, and then they're all going to be on the right, on the other side. So they're just, it really depends on which way you have your model line down. I always put my nitrogens on the left side, and that way I don't know, but you never, I mean, you can't tell if you're looking at a picture, you wouldn't be able to flip it over. So you just know it's between a carbon and a nitrogen, and that carbon has that double bonded oxygen. Okay, so this is a unique molecule. It has rings made of carbon. Oh, very good. Steroid, yes. Okay. Now look at this one. It looks very different from this. Okay, steroid is a lipid. Lipids have very little oxygen. The molecules are all black and white because they're made up primarily like 95% carbon and hydrogen. And you get a tiny, less than 5% of oxygen. Okay, so triglycerides, steroids, black and white. You get, you get to something, a model that looks like this, and we have a whole different ratio, right? Because we've got red, white, and black. So we know that this has to be something else. So it says this ratio would suggest this molecule is what about this ratio? Carbohydrate and not a lipid. Because what is the ratio in carbohydrates? What percentage is carbon? Yep, carbon's 25%, hydrogen's 50%, and oxygen is 25%. So that ratio is just very, very different between carbohydrates and lipids. So this is a molecule, like I said, we didn't go over it a whole lot. It forms a backbone. It is the molecule, we just say you just have to learn this one. It has 3 carbons. Yep, it's glycerol, okay. And once you attach 3 fatty acids to it, it becomes a triglyceride. Yep. Okay. This is the element phosphate. So we know that this is the organic molecule, and you can just give me 3 letters because it's ATP. That's all we really care about, because you'll go into ATP a little bit this semester and then a lot more next semester. So we just leave it in lab, and we do so much with the other ones, okay? You don't have to. This is the nitrogenous base, this is the pentose sugar, and then these are the phosphate groups, but that won't be, you won't have to know that. Okay, so this is how we lay out a fatty acid. Okay, I didn't have a picture in the last PowerPoint. So this is a great presentation of a fatty acid. This is the carboxyl group. It's the functional group. And this is called, I don't know why, but we call this the head. And then this is the fatty acid tail. When you are looking to see if it is saturated or not, you work your way along and you see if each carbon has 2 hydrogens, okay? And you just kind of go and you go and you go, huh, well, that's interesting, because this one doesn't have 2 hydrogens, this one doesn't have 2 hydrogens, because there is a double bond here. If there is a double bond here, is this a saturated molecule? Yes or no? No, it is unsaturated. Very good. Okay. Say this is mono unsaturated because it has 1. Sometimes you'll see 2, but I think all of our models just have 1. And we do try our best to represent them this way, where we try and put them relatively in the middle, and we try and present them so that you would see both the double bond and that you would see that you're missing a hydrogen here and a hydrogen here. You know, we try not to be sneaky. We do realize you aren't physically in lab, so we do try and present them, you know. Okay, so what is this monster of a molecule? A triglyceride, yep. And so again, you can kind of work your way along, and you can see, so for this guy here, is it saturated or unsaturated for this first one that's close? Correct, it's saturated. And then, I don't know if you guys can tell on this one, if you work it your way through, this is a double bond, so it would be unsaturated, yeah. And like, we wouldn't use this one to call it out because, I mean, it's not ideal to see. It's just, I'm just using it as an example, okay? All right, but it does say when this molecule was formed, and then from its 4 components would be glycerol plus 3 fatty acids, how many water molecules were released? 3. How do you know if it's unsaturated? So an unsaturated fatty acid has a double bond, which is a little hard to see in this one. So in this one, it's a little bit easier to see. If there is a carbon-to-carbon double bond, it means that this fatty acid is unsaturated, because instead of having as many hydrogens as it could, it's chosen to double bond to share those electrons with carbon instead of with hydrogen. So this, having a carbon-to-carbon double bond, means that it's unsaturated, as compared to on this first one, every single carbon has 2 hydrogens. So it doesn't share any electrons between carbons. So that's saturated. yes, so this is where dehydration synthesis occurs. Okay, so this molecule was formed from how many monosaccharides? Oops. 2, yes. So what is it known as? So monosaccharide plus monosaccharide forms a disaccharide. Okay, so I'm just gonna go over this slide and then I'll go back. Okay, so this, if we just did one of these, so just ignore this one here. Okay, so if we just did this one, this would be the monosaccharide called what? Specifically by name. They built it in the video, kind of. We would have built it in class. No. So alanine is an amino acid. So this would have been glucose. So if we just did this one here, this is glucose. Okay. Then if you did glucose plus glucose, the whole thing, this would be known as maltose or disaccharide. So I'm not sure which one it would ask for on like a lab exam. You should know that this is known, that if you see this structure, I would think that it would be reasonable to ask you that this is glucose, okay? And we're not going to ask you galactose, okay? Like I said, they're very, very similar. But I would think that the answer choice is going to be more likely than like monosaccharide, okay? But if you see both of these, like as it is in here, it's probably going to be like, if you saw this question, identify this organic molecule, it's probably looking for disaccharide. It might be asking for maltose, but probably not. It's probably looking for disaccharide, but either of those would be correct. But your lab exam and your lab quiz are both multiple choice. Okay. Okay, so I'm gonna go back to the previous one. Maybe, I think I am. There we go, okay. So on this slide, your question, so this gets more into kind of what we cover in lecture. The 3 arrows indicate the sites of dehydration synthesis. Okay, so that is going to be true. Each of the 3 indicated oxygen atoms is the remnant of the interaction between a hydroxyl group and a carboxyl group. Is also true, I believe. I don't know if I have that one on my thing. Yes, yeah. So we didn't cover exactly like which molecules were involved in each of the reactions for the fatty acids in glycerol. So what you do need to know is that when fatty acids attach to glycerol, that again, it's dehydration synthesis and it produces a water. If we were to break this triglyceride apart, you would release each of these fatty acids, but each of these would consume a water to release them. So we don't, like I said, you don't have to know exactly which of the molecules are participate in each reaction step. Okay. Oh, so this one we just went over. So identify this organic molecule. So C6H12O6, also known as glucose. Yep. So again, this is not something you would need. This one would be probably obvious which one is involved in dehydration synthesis. It would be this one, because this is the one that's used to form the oxygen bridge, right, the middle one. And the only thing I do want to point out is, like I said in the video, he forgot to leave this red oxygen. So just make sure that you do know that this oxygen is left, because it's the oxygen that forms the bridge between the two monosaccharides. It's, I mean, not a big deal, but it is kind of a big thing that they are linked by oxygen. They're not, it's not carbon to carbon. You do leave that oxygen there. Okay, so this is, again, so this is a glucose, this is a glucose to form the disaccharide maltose. So glucose plus glucose would form a disaccharide known as maltose using the process of dehydration synthesis. Okay. Glycerol plus what component? You need 3 what to make a triglyceride? They can be saturated or unsaturated. Fatty acids. Yep, so glycerol and 3 fatty acids through dehydration synthesis. So it's that top arrow, make a triglyceride, and 3 waters. Don't worry about, again, we're not going to do ATP. Amino acid plus another what? Amino acid, very good. Through dehydration synthesis is dipeptide plus H2O. And then the back arrows represent, so forward is dehydration synthesis, back arrows represent the process of what? Hydrolysis. Okay, so if you make something, it's dehydration synthesis. If you break something apart, that's this back arrow here, it's hydrolysis. Okay. So which arrow below, 1 or 2, indicates the process of hydrolysis? Arrow number 2, very good. True or false, steroid molecules are lipids. You have 50-50 chance. True. Steroids are lipids, so are phospholipids, and so are triglycerides. Okay. Sucrose is a monosaccharide. False. It is a what? That's right, it's a disaccharide. Proteins are large polypeptides. Just guess on this one. It is true, that is correct. Water is released in hydrolytic reactions. So is water released or is it consumed in hydrolysis? Water is consumed in hydrolysis, so this one would be false. Water is released or produced in what type of reaction? That's right, dehydration synthesis, so the other one. Okay. A tripeptide contains 3 peptide bonds. False, because it only contains 2. Very good. Oh, and that's it. Okay. So, so that's the end of the slideshow, and actually the end of lecture too. So, this topic is actually on today's quiz. So the quizzes, if you go over the remote learning schedule, it will have the quiz topics. And so, and I don't, so they're set by the department. The quiz topics that we cover each week. So for this week, we cover, you have 5 questions on the microscope, and then you have 5 questions on organic molecules as well. So that's why I leave it open until 10 o'clock tonight, because I don't want you taking it like right now, because you probably want to, you know, review a little bit before you take it. You know, and I do try and review as much as I can before you take your quiz. So it's on last week's, and then it's on this week's. And then in 2 weeks, you'll have another quiz, and it'll have 5 or 6 questions on histology, which is next week's topic. And then it'll have 4 or 5 questions on the axial skeleton, which is that topic of that day. So you might want to spend a little bit of time also studying, you know, axial skeleton for that day, if you don't have as much time that day to study. So the quizzes are usually on 2 weeks. There's one quiz that we just have on, I think, 1 week topic. So it's on, yeah, so it's on last week's and then on today's. And then the lab exams are on either, this time it's on 4 weeks, and then it's 3 weeks, and then 3 weeks. So are there any other questions? Okay, so if you are comfortable, then you guys are dismissed. And then if you want to ask me other questions, you know, I will hang out. So thank you. You have a good day too.